Good afternoon, and welcome to the USDA Human Nutrition Research Center on Aging's Monday seminar series. Before introducing today's speaker, let me remind you to ask your questions for our speaker using the Q&A icon located on the webinar toolbar. Please add your questions as they arise during the presentation. Let me welcome today's speaker, Dr. Stefano Monti. Dr. Monti is a computational biologist at Boston University in the section of Computational Biomedicine, with joint appointments in the Biostatistics department and the Bioinformatics Program. Dr. Monti received his PhD in Intelligent Systems and Artificial Intelligence from the University of Pittsburgh and completed his training with a postdoctoral fellowship at the Robotics Institute at Carnegie Mellon. His doctoral and postdoctoral research focused on the development of machine learning and knowledge discovery methodologies, with a particular emphasis on the probabilistic reasoning and Bayesian approaches to modeling biomedical data. Since 2001, he has worked in the field of cancer genomics, first as a research scientist at the Whitehead Institute's Center for Genomic Research, and later as a computational biologist in the cancer program at the Broad Institute. Dr. Monti's laboratory integrates systems biology and machine learning and bioinformatics approaches to investigate the molecular drivers of human disease with the goals of advancing prevention and care. This multidisciplinary effort relies on the development of novel computational methodologies, on the design of experiments based on the generation of integrative analysis of high-throughput multi-omics data, with the goal of identifying novel therapeutic targets and developing accurate diagnostic and prognostic biomarkers. His current research includes the study of molecular mechanisms of tumor initiation and progression, and the role played in it by environmental exposures, as well as the study of biological factors contributing to healthy aging and extreme longevity. And today, Dr. Monti will discuss this research in a presentation titled Multi-Omics Modeling to Study Cancer and Healthy Aging. And with that, let me turn the mic over to Dr. Monti.
Thank you very much, Paul. Thank you for the invitation, and thank you for whomever is out there in the ether. I cannot tell if I'm talking to myself or to real human beings, but here I am. So yeah, the title is pretty general, multi-omics modeling to study cancer and healthy aging. And Paul did already a good job, a good job at telling you a little bit about what my lab is about. And so I will not repeat it. But so basically, my lab has a dual focus. Basically, we are interested in investigating both how to investigate human diseases in order to advance caring, advance targeted therapy. Basically, we do this through the use of in silico biology approaches to investigate mechanism of action towards the identification of therapeutic targets and towards the development of diagnostic, prognostic, and therapeutic biomarkers. On the other hand, we're also very much interested in using omics-based in silico biology to advance prevention. And prevention that can be thought of as early detection, but actually really even more so really preventing the disease from arising in the first place. And a lot in the past in my lab was focused on actually modeling exposure, environmental exposure. More recently, in the last few years, I shifted more my focus to study healthy aging and how basically the protective mechanisms associated with healthy aging might actually be leveraged to advance care and to advance the avoidance of disease. And so in both pursuing both these goals, the common thread, the common unifying factor is basically the development of computational methodologies and tools that basically support in silico biology. So I mentioned silico biology. Let me briefly give you my interpretation of what it means, and which will also give you a sense of our research approach. So our main focus is the understanding of the phenotype, right? The phenotypes that could be the disease, or it could be actually not the disease, the healthy aging, as I mentioned. and how this is connected to the underlying biology, to the underlying molecular machinery. And we hope that the analysis of paired variation in both the phenotype and the underlying biology will help us establish meaningful connections, associations, correlations between the two, hoping in turn that this will lead to insights into the causes, the mechanism. So contrary to conventional wisdom, we're using correlation to suggest causation all the time. Of course, we have access to model organisms and experimental approaches that allow us actually to validate the suggested causes coming out of the association analysis. And so to quantifying the underlying molecular machinery that quantify the variation in the underlying molecular machinery, we rely on so-called high-throughput omics assays. They query the molecular machinery at different levels, the genome, the transcriptome, the epigenome, the proteome, and so forth. And this, of course, the analysis of these omics data support discovery, okay? So in a nutshell, here's how many of our studies proceed. We have a question or a hypothesis of interest in mind, which leads us to the design of experiments, where the experimental condition could be biospecimen from a population of interest or perturbations, molecular or chemical. We then extract and profile the molecular entities of interest, right? This could be genes, transcripts, proteins, and so forth. And basically this go on to fill in large matrices. Think of them as huge Excel spreadsheets, measuring thousands, if not hundreds of thousands, if not millions of molecular features in multiple experimental conditions. And the analysis is about establishing connection between the rows, basically between the molecular features, between the column, between the experimental condition, and between the rows and the columns. Okay, so, and then of course, this analysis loop can continue, and the discovery supported by the data analysis can lead to the new experiment. So here in a nutshell is how many many of our studies are done, right? And so perhaps one of the best known and early examples of this multi-layer, multi-omics study is the so-called TCGA, that's a Cancer Genome Atlas, where thousands of tumors from more than 30 tissues were profiled at multiple levels, genome, transcriptome, they're listed there basically. And all data are now publicly available for research. And there are many more examples. We started in the late 90s, early 2000, thinking that there were never enough data. Now, basically, we really are getting to an age where actually we are living in a wealth of data that we can routinely use for in silico validation and discovery. But so TCGA was an example. Perhaps an example closer to home are the multiple consortia studying healthy aging and longevity, all based on the collection and multi-omics profiling of biospecimen from subjects spanning a wide range of ages, including so-called exceptional longevity, or EL individuals for short. These are multi-institutional projects which we are part of, and involve collaboration with Paola Sebastiani at Tufts, Tom Pearls of New England Centenarian Study fame, Nick Shork at TGen, and a host of other collaborators across the nation and also in other countries. And so the specific aims of all these consortia have differences, but the common thread is the study of healthy aging. That is, understanding the contributors to extending a person's health span, okay? So I talked about cancer and aging, and so, sorry, I forgot about this, that actually one of the studies is actually integrated longevity omics, ILO for short, which is a very cool study that I invite you to Google, for, and involves a collaboration with Thomas Pearls, Paola Sebastiani, Nick Shork, Vera Gorbunova, and Rich Miller, University of Michigan, and many others. So, so why cancer and aging? You might think that these are different domains, And many of you are probably familiar with the concept of hallmarks. Hallmarks have been defined to try to conceptualize and organize the complexity of these phenotypes of cancer and aging into arguably evolving sets of underlying principles, underlying rules. Basically, a set of biological and phenotypic rules that govern the malignant transformation on one end and the aging process on the other. So hallmarks such as genomic instabilities and replicative potential, limitless replicative potentials were among the first hallmarks of cancer that had been identified, to which over time, many more have been added, including cell plasticity and polymorphic microbiomes more recently. In aging, hallmarks include, again, genomic stability, to notice the overlap, and then others that are unique to aging, such as mitochondrial dysfunction, chronic inflammation, and so forth. So what is important is that some of these hallmarks are shared by the two phenotypes. And as I mentioned, genomic stability is one of them. While some others, for example, aging hallmarks, are likely to suppress oncogenesis and can be thought of as antagonistic hallmarks. And there are other connections. But this is all to say that studying one has bearing on understanding of the other. And so while our lab's investigation of the two started as these independent endeavors, we're now moving towards investigating the interplay. Okay. So in the rest of the talk, I will present some vignettes from ongoing studies. And I apologize, some of them will be squarely focused on cancer, head and neck cancer in particular, or breast cancer. And they are aimed at advancing therapy and prevention, here mostly thought of as, I apologize for the song, mostly thought as early detection. But actually, some of them will investigate the interplay between cancer and aging. If time allows, I will give the audience a brief overview of a breast cancer study that uses some of the same assay and methodological tools, but instead of studying the interplay with aging, it focuses on understanding the role of metabolic comorbidities, insulin-resistant type 2 diabetes in particular, on cancer aggressiveness and metastasis. And then I will conclude with a slightly more detailed description of a metabolomics-based study of aging and exceptional longevity in healthy individuals. So the common denominator in all these studies, beside the aforementioned yin and yang of aging and cancer, is basically the use of shared tools of in silico and computational biology. Okay, so let's start with head and neck cancer, and we in particular with head and neck cancer of the oral cavity, primarily presented as oral squamous cell carcinoma, or OSCC for short, which is the most prevalent type of head and neck cancer, is the 7th most common cancer in the world, with close to 1 million new cases per year worldwide. The 5-year survival rate is not great, and actually it reduces to 50% for advanced metastatic tumors. It is linked to alcohol consumption and tobacco use. And unfortunately, its molecular mechanisms are only partially understood, and few effective biomarkers and treatments are available. Basically, the therapeutic intervention has remained unchanged more or less for the last 30 years, if you don't factor in basically advances in robotics assisted surgery. So OSCC, oral cancer, arises from premalignant lesion in the oral cavity through a multi-step series of clinical and histopathological stages. And early detection here is key to increasing long-term survival. The earlier the detection, the better is your prognosis. And so today we'll present highlights from few ongoing studies, and as suggested by introduction, we'll emphasize the in silico aspects of the studies. And so I will start with the first study, which is mainly a shout out to Eric Reed, who actually is a graduate of our lab, and now is a research scientist at Tufts DISC. And so Eric led a very elegant multi-step analysis aimed at elucidating the mechanisms of pharmacologic inhibition of Wnt beta-catenin CBP, which is a promising candidate therapy in multiple cancer, including head and neck cancer. So perhaps many of you know that Wnt beta-catenin is a very important signaling pathways. having a role in normal biology, but when aberrantly activated, it has been shown that to be associated with cancer stem cell-like properties and aggressiveness, and including also resistance to treatment. So in this study, we started by analyzing publicly available single-cell data sets of head and neck cancer, and we used a newly developed methodology to organize the cancer cells from multiple patients into a taxonomy, identifying cellular states with distinct molecular programs, some of which were shown to be significantly associated with disease-free and overall survival. We then projected these states into a lower dimensional space and performed causal network inference, which showed a novel connection between beta-catenin activity and partial EMT through activation of mTORC1. And this signaling cascade was then functional validated in independent data set, the Cancer Genome Atlas and the Cancer Cell Line Encyclopedia, some of the data set publicly available that I mentioned to you before. So partial EMT is of particular interest because it represents cellular states where epithelial cells manifest characteristic of mesenchymal cells, but do not fully undergo the transformation, and have been shown to be early predictors on other metastases. We went on to basically validate this signaling cascade, this causal signaling cascade going from beta-catenin CBP to mTORC to a PMT through in vitro, and in vivo experiment. So to summarize this finding, basically driven completely by in silico analysis, we identify this signaling cascade going from beta-catenin CBP to mTORC to PMT. We have validated in vitro and vivo these findings, and the translational significance is that pharmacologic inhibition of beta-catenin CBP is emerging as a promising head and neck cancer therapeutic intervention. Another shout out to Eric is basically downloaded from GitHub. And this tool we use routinely in multiple studies, and since that study has been used in others, in particular one led by another graduate of our program, and now a scientist at Tufts, Tanya Karajanis, which basically perform an extensive single set study of primary peripheral blood mononuclear cells collected from centenarians and control, and recently published. We were using it also in multiple other studies, which I don't have time to talk about right now. So the second study I want to mention is motivated by our interest in investigating the molecular mechanisms driving progression of pre-malignant lesions to oral cancer, and the potential role played in it by the oral microbiome. Okay, so the oral microbiome is the second largest microbiota in the human body, and changes in its composition have been shown to be associated to oral cancer. So we took a profiling approach that allowed us to quantify both the host and the microbiome as well, and to begin to look at their crosstalk. Okay, so we performed so-called total bulk RNA-seq profiling on tissues from the oral cavity of 66 patients. And total RNA-seq supports a two-step approach where first the reads are aligned to the human genome and used to quantify the host gene expression, and then the remaining reads, the subtraction, if you wish, are aligned to libraries of microbial genome to identify the presence of microbial species in the samples. So as I said, we performed total RNA-seq profiling in a set of 66 samples of oral tissues from as many patients, representing different types of pre-malignant and malignant lesion, as well as controls. And this was made possible by collaboration by wonderful surgeons at Erie County Medical Center and Brigham Women at Miami Cancer Institute, Jennifer Pristina and Alessandro Villa. So what did the analysis of the data generated tell us? Well, Not surprising, if we do hierarchical clustering, we observe considerable transcriptional heterogeneity within and across histological groups, histopathology groups. We saw the controls and oral cancer were very distinct, and the pre-malignant lesions covered a gradient between the two, with dysplasia closer to the cancer group, and another type of pre-malignant lesions called hyperkeratosis closer to the control, although with exception. Next, basically, we did other in silico annotation. Basically, we annotated a sample based on their enrichment for several malignant transforming metastatic signatures. This included a previously derived transcriptional signature distinguishing malignant transforming versus non-transforming. Transforming versus non-transforming. OPMDs, which is short for oral potential malignant disorder. Then a cancer versus control signature derived from the TCGA that I mentioned before. And then a previously derived signature of partial EMT, that phenotype that I mentioned to you that is being associated with cancer progression. And all this signature basically showed a monotonic increase in enrichment as we move from control to cancer through the premalignant lesions. Also, differential gene and pathway analysis across groups highlighted enrichment of oncogenic and immune pathways in premalignant lesion. As you can see, some of the same pathways were enriched in the cancer group and also in the PML groups, although more moderately and mostly driven by the dysplastic tissues. What about the microbial profiling? Here we relied on collaborations with the lab of Evan Johnson, who used to be at BU and now is at Rochester. Sorry, I'm blanking out the name of the university. Rutgers University, apologies. So the microbial profiling showed a monotonic increase in alpha diversity and an increased abundance of selected microbes and actually fungi as we move from control to premalignant to oral cancer. And some of these microbes have been shown previously to be associated with oral malignancies, such as Fusobacterium and Candida. And also leveraging bioinformatic tools, we were able to connect the host transcriptome to the microbe and identified a significant association between oncogenic and immune pathway and microbial species more abundant in PML and oral cancer than in control. So altogether, basically, this study told us that premalignant lesions are a very heterogeneous set of lesions, are enriched for malignant transforming and partially EMT signature, and also for oncogenic and immune pathways, and have a significantly higher level of immune activity, although in premalignant lesion, whether this represents response to the insult or actually is a contribution to the progression cannot be answered by this data. Also, we show that differentially abundant microbial species in PML and oral cancer are associated with pathways that promote malignancy and immune activity. And the translational significance, again, is that this data and this analysis present the potential for developing predictive markers of malignant transformation risk. Basically, can support early detection. Okay, so bear with me. These were studies that were focusing completely on cancer, no association with aging. The next one that I'm telling you about is trying to explore this interplay between aging and OSCC, and basically thinking of age as a comorbidity of oral cancer. And these are a collaborative study with the labs of Bob Varelas and Maria Kurudzinska, who are also at Boston University, and to study the interplay between aging and cancer, we're adopting both in silico approaches in my lab and functional approaches in their lab. So Since I'm not yet at liberty to share the result of the functional approaches, I will focus here on the in silico studies we're conducting with Lina Curling, which is a talented bioinformatics trainee in our lab, leading this analysis. And the study is primarily aimed at investigating age-associated shifts in intratumor heterogeneity. Eventually, of course, this result will be integrated with the result of the functional studies by the other labs. So aging is a risk factor for many cancer types, as you can see from the plot on the left. So the aging risk, the cancer risk increases unless you make it past 85 and, or past 80 or so, after which actually your risk decreases. And older age is associated with worse survival, okay? Of course, these curves are adjusted for age just to make it clear. So definitely your age-adjusted survival as you grow older gets worse. So what leads to a worse overall survival in old versus young people? What are the causes? So here we are asking questions about tumor heterogeneity. So are there distinct cell populations that are more prevalent in old versus young patient tumors? Is there altered signaling occurring in old versus young patient? These are the questions that we're trying to answer. And to do those, to begin investigating this question, we took a data-driven approach. Namely, we collected all the head and neck single-cell RNA-seq data set we could find and combined them in a well-integrated and age-annotated data set. This is, to our knowledge, the largest available single-cell data set of head and neck cancer patients, profiling more than 230,000 cells across 54 patients with ages ranging between 30 and 89. And we will continue updating this resource as new studies become available. So the analysis proceeded by first identifying the major cell type compartments, the epithelial compartment, the immune compartment, so forth, and by then refining cell type annotation within compartments. And then, of course, associating the cell types with the age of the patients. Okay, so what we did was really performing, based on generalized linear model, we performed differential/regression analysis to connect cell type proportion shifts with age. And we identified several cell types, both in the immune and non-immune compartments, whose relative abundance significantly increased or decreased with age. I don't ask you to parse this plot that are very busy, but I want to bring you to some highlights. In the immune compartment, naive and cytotoxic CD8 T cells were more abundant in young than in old patients, and so were myeloid cells, while Tregs and quote-unquote dysfunctional CD8 T cells were enriched in old. I'm saying quote-unquote dysfunctional because we're using a signature of dysfunction that is not necessarily really predictive of dysfunction. It might simply reflect activation of certain markers such as CTLA4, CTLA, PD-1, and so forth that have been associated or have been shown to be associated with dysfunction, but they do not necessarily have that role. In the non-immune compartment on the right, what was really jumping out was basically an enrichment of CAFs, so-called cancer-associated fibroblasts, in old than in young patients. So together, these results suggest that on one side, a more active and functional immune environment in young than in old patients. And as far as the tumor microenvironment is concerned, in older patients, we see an enrichment for CAS, for cancer-associated fibroblasts, suggestive of extracellular metric stiffening and fibrosis, both of which are associated with EMT. EMT is basically epithelial mesenchymal transition, is a program that is often associated with tumor progression and metastasis. We also performed differential regression analysis to connect, not differential analysis. We performed cell-cell communication analysis to look for age-associated patterns. And we identified several ligand receptor pairs whose activity was strongly associated with age. Fortunately, I'm not at liberty to share the details of this finding in terms of what actually specific ligand receptor we identified, but basically we noticed, for example, an increased signaling from macrophages to immune and vascular endothelial cells in all patients, as well as actually signaling from fibroblasts to the vascular endothelial and epithelial cell in all. We also observe a more ubiquitous, not age-specific signaling between epithelia midkinds and immune cells. So there is a lot to parse in this data, and we're in the process of actually validating some of these results with functional assays, and a manuscript is in preparation. But another point that I would like to bring to your attention is that actually this single cell atlas that we're developing and we plan to submit for publication is a resource that we feel can be useful to the larger research community. And it can be used to interrogate other phenotypes, to interrogate other programs, molecular or clinical phenotypes that you might be interested in, and looking for association between that phenotype and shifts in cell type proportions. So let's see. I think I'm going to skip, in the interest of time, the breast cancer and metabolic dysfunction vignette, and I'm going to jump. You see, I had this safety button here, depending on how long it would take me to get here. So I'm going to skip and go into the next. If it let me. It doesn't look like it lets me do it. Okay, I'll skip the slides. And I will focus on the last but not least of the studies that I want to feature, that I want to emphasize here. And this is also focused on age, but this time is not in the context of cancer. I'm now shifting to describe our in silico studies of healthy aging, in particular now uses of metabolomic profiling to study aging processes. And so here I bring back the slide that I featured at the beginning, where basically we are part of multiple consortia that are profiling, multi-omics profiling plasma and other, in human only plasma, from subjects spanning a wide range of ages. And in particular, in this study that we'll highlight now, I'm focusing on a cohort of patients drawn from the New England Centenarian study, led by Tom Perls at Boston University and by Paolo Sebastiani at Tufts. And so metabolomic profiling has emerged as a valuable tool in aging research. It offers valuable insights into the aging process at molecular level. And the hope is that it may support the development of targeted intervention, as well as actually lifestyle changes that might promote healthy aging and mitigate age-related diseases. So in this study, which is ongoing, we aim to investigate mechanisms, but actually more accurately to investigate correlates of aging and exceptional longevity. EL, I will use this acronym often. And we want to do this through the analysis of metabolic profiles of blood serum from a cohort of individuals spanning ages, very wide age range. We're doing so by focusing on a data set that is highly enriched for long-lived individuals, so-called centenarian, supercentenarian. And we will focus primarily on distinguishing between aging and EL signals. And we will do so by comparing, contrasting, and integrating across multiple independent metabolomic data sets. As I said, our main focus is the New England Centenarian study, but as I also mentioned before, we will leverage other data sets to which we have access to basically integrate with the results from our study. So the rationale, of course, is that, as I mentioned, we're using correlation to suggest causation all the time. So we will use association between age-associated phenotypes and metabolites levels as the first source of evidence to identify candidates for intervention. We're not making any claims about causality, cause and effect, but we're simply hoping that by identifying this association, we can help bump up the priority list, the most likely candidates for intervention that, combined with other sources of evidence from independent studies or from animal experiments, will be the potential focus of developing new therapeutics or lifestyle changes. So in our study, as I said, we focused on a cohort from the New England Centenarian study, which is one of the largest studies of long-lived individuals in North America, again, led by Tom Pearls. We integrated the results from this data by contrasting with other metabolomic studies that did not include EL subjects. So comparing the EL enriched with the non-EL enriched data set help us distinguish the two. And then by also validating some of the results in independent data sets that also include EL subjects. These were the famous LLFS, short for Long Life Family Study, led by Mike Provence and including Tom Pearl, Paola Sebastiani, myself, and a host of other investigators. And also we compared to a Chinese study, compared long-lived individuals to their offspring in more than 100 Chinese families. So here's a quick recap of the studies. They differ in the assays used for doing metabolomics. Some use metabolome, some other use untargeted mass spec. They differ in the number of metabolites profiled, in the number of samples, and in the age ranges. And here I'm giving you the quantiles and this is something important. Even the other centenarian studies, namely LLFS and the Chinese studies, do not reach the extreme ages obtained in the next cohort, right? You can see that the 75 percentile is 103 for next, while it's in the 90s for the other two centenarian. So this is very important. So a similar analysis was performed on a cohort from the Long Life Family Studies, and a manuscript led by Paola Sebastiani is under review. So why repeat it here? What distinguishes this study from the LLFI study? I already alluded to some of the aspects. The LLFI study provided a large number of subjects, but a relatively smaller number of metabolites, about 400. And while it focused on families with long life or disposition, it included few centenarians and supercentenarian. On the other hand, the New England Centenarian study that I'm going to discuss today profiled fewer subjects, around 200, but assayed a significantly larger number of metabolites, more than 1,000, and the cohort was highly enriched for exceptional longevity subjects. So the next cohort really allowed us to focus on the EL phenotype. Here's the age distribution in NEXT compared to LLFS. You can see the further skew to the right for the next cohort. And so the question I will address, the scientific question I'll address, also dictated the statistical models that we used. So we regressed metabolite levels on aging to identify age-associated metabolites. We performed differential analysis between EL and non-EL subjects to further focus on EL individual only. And since we have follow-up information in the individuals past the collection of blood, we can test for association between metabolite levels and length of survival. So a bird's eye view of the data set tells us this is principal component analysis, and you can see basically that each dot represents subjects, and you can see that basically EL subjects segregate from offspring and control. Also, if we look at the variability of single metabolites, we can see that their variability increases at the extreme, at the 2H extreme, something that has also been observed in proteomics profiles in a previous study. The signal from the aging and the EL phenotypes is quite strong, as shown here, with more than 1/3 of the metabolites assayed significantly associated with age and or EL status. And the overlap between the two signatures is considerable, a point to which I will return. Another way of showing the strong signal from the two phenotypes is by plotting the heat map, which also point to the fact that the number of metabolites going up with age or EL status is considerably larger than the metabolites going down. And of course, volcano plots can show and highlight specific metabolites. But as I said, the number of metabolites is very large. So I will not go over single metabolites here, but I will give you some summary that hopefully will convey some important insights. So the first thing that I want to notice is that the results and the metabolites that are differentially expressed recapture largely what's already known, right? Basically, we can check all the marks with respect to a recent review, cataloging the main metabolomics collective findings throughout the years, with the exception of lipids, which were not assayed in our metabolome platform. But of course, what we want to go is beyond what's already known, right? And so in particular, we want to focus on what's unique to AL subject. And to that end, earlier I emphasized the larger overlap between the two, but actually what we really care about is what's unique to the two, which is basically the off-diagonal metabolites, if you wish. But ideally for this comparison, so this would be basically the centenarian or EL only markers, because they were found in the EL versus not analysis, and these are the age only markers, the ones that were found in the age analysis but not in the EL analysis. But ideally, to do this analysis properly, we would need to have access to data sets that do not include centenarians. Otherwise, also the age signature here might be actually enriched for centenarian markers. And so to do this, actually, we integrated multiple data sets. So on the one hand, we use the signature of centenarian versus not from the New England Centenarian Study. But then actually, thanks to collaboration with the Institute of System Biology in Seattle and with Luigi Ferrucci at the NIA, we had access to other sources of data sets, the Hariri and BLSA Baltimore longitudinal study of aging, which gave us access basically to information about age trends, metabolite age trends in population not inclusive of centenarians. And basically what we did was really a simple set difference of the two, slightly more rigorously done, but that's really basically conceptually what we did. And so that allowed us to identify EL-only markers and age-only markers, so to speak. And here I'm showing you some prototypical expression or metabolite level patterns of what I mean. These are so-called centenarian-only markers. These are basically markers that do not change with age, but then have a significant change in centenarians, either up in centenarians or down in centenarians. And these are examples of what I like to call bucking the trend metabolites. Basically, there are metabolites that manifest a clear trend with age, either down or up. But these metabolites do not behave consistently in centenarians. Okay, so, and actually tryptophan is one of, even though not as significant in other, is one of these metabolites that bucks this trend. And so what we did, basically, we derived a lot of signatures, metabolite signature, and still there is the issue of how to make sense of this. So first of all, we focused on specific signatures that are more relevant to investigating the EL phenotype. So the age-only signature, the EL-only signature, and the survival signature. And then basically we used annotation by enrichment analysis to try to group multiple metabolites into biological processes, into biological categories that might be actually easier to interpret. So even when reducing to metabolite classes, as I'm showing here, basically these are the different signatures. These are different metabolite categories that were shown to be enriched in the different signatures. You can see that there are a lot of results to parse. But so what we did actually was try and cross-examining or triangulating between the enrichment of the signature of aging and longevity and the signature of survival, which allowed us actually to focus on a smaller set of metabolite sets that seem to be particular interest. So the set of metabolites were from two main classes: bile acids, which were enriched in EL-only signatures, and higher level were associated with better survival. And then C19 steroids, which include the androgenic, the pregnenolone, and progestin steroids, which are down with age, but that also higher level are associated with better survival. Okay, why these two different type of tests is because given the relatively small sample size, 200 or so patients, we did not have enough strong signal to identify single metabolites associated with survival. But when we grouped into categories and performed rank-based enrichment, we were able to find significant associations. So, and these are basically for example, the bile acids that drive the association with survival. So in summary, we identify multiple categories of metabolites that were either enriched in the centenarian or enriched in the age-only signatures. Some that were going up, such as the bile acids, such as they were going down, others that were going down, such as the steroids, and some of which were actually associated with improved survival. But actually, the higher expression was associated with the better survival. For others, the lower expression was associated with survival. And this allowed us to actually hypothesize that some of them are actually really markers of exceptional longevity. And this includes bile acids and androgenic steroids, androgenic progestin and pregnenolone steroids. But there are other actually that are more likely to be end-of-life markers. Basically, they're more abundant in centenarians, but actually their higher abundance is associated with worse survival, which suggests that it might represent simply markers of end-of-life. So digging deeper and analyzing in a multivariate fashion the relationship among multiple metabolite classes, a story is emerging where the de novo NAD synthesis pathway is closely interacting with the kynurenine and aspartate pathways and point to a potential role played by the gut bacteria metabolism in aging and enhanced survival. And this is actually work done in close collaboration actually with the necessary input from Michael Lewis-Garden, who's also a scientist at Tufts, and who has a very extensive understanding of metabolism. So in summary, deep metabolomic profiling we performed deep metabolomic profiling, one of the largest EL-enriched data sets, which confirmed and expanded upon previous finding. It pointed to the increased secondary and primary bile acids as significantly enriched in EL only, in exceptional longevity subjects, and a decrease of steroids that was significantly associated with the age, but not with EL subjects. From the methodological point of view, I want to highlight basically that actually our strategy of looking at metabolite classes rather than single metabolites helped us increase the statistical power of our analysis, and triangulating between aging, extreme longevity, and survival enabled us to distinguish what is the exceptional longevity signal from the end-of-life signal. And I think I will stop here in the interest of time because I'm past the 15 minutes. And I just wanted to thank the many collaborators we had in these studies. Of course, when it comes to aging, Paola and Tom, Paola Sebastiani at Tufts, now at Tufts, used to be at Boston University, Tom, still at Boston University, Michael Lustgarden, as well as Tanya Karajanis and Eric Reed, and many others that were essential to this project. When it comes to the cancer studies, these were more Boston University Center, and I want to thank the labs of Maria Kurudziska and Bob Varelas, as well the members, past and present of my labs. And well, I didn't have time to touch upon the work on breast cancer, which is done in collaboration with Gerald Dennis. Sorry for the spelling, and Daniel Rowe, who's a surgeon here, and other talented trainees. And I'll stop here and I'll take questions if there are any.
Well, there are. Thank you very much for presenting some very interesting observations and really giving a clear explanation of a very complex subject. And with that, I'll go to the first question, and that comes from Larry Parnell. And he states that some of the largest mammals, like elephants and whales, have been reported to have very low rates of cancer. This is also true for the naked mole rat. And so his question is, have you seen in your research any sharing of cell type, genetic, or metabolomic signals from the studies on these other mammals?
Okay, this seems like a planted question, because it's very much what the Integrated Longevity Omics project is about. As you can see from, I included this because I really love this this image. Part of the Longevity Omics project, which is led by Nick Short, is focused on actually doing multi-omics profiling across species, including the naked mole rat, including long and short-living animals, species. And so we do not have yet strong results from the cross-examining of these different species, but that's where we're going, basically. The idea is actually to perform paired omics profiling in these species, and then looking for commonalities and differences, and see if actually one can inform the other. So right now, no, I don't have conclusive results on that analysis, integrative analysis, but in the future, this will be an important part of this and other studies.
The next question comes from Joel Mason, and his first comment is that that he won't pretend to understand all of the strategies by which you integrate the various omics databases, but it does seem that the methods only allow you to establish associations, not causality. His question is whether there is some analysis, perhaps analogous to Mendelian randomization, that might enable you to begin making conclusions regarding causality.
Absolutely, very, very good question. And I first want to point that even without Mendelian randomization, there are techniques that tries to move a step further from simple correlation, even when you're using only observational data. And this is the use of Markov network, Bayesian networks, and so forth. But still, the point is valid and is well taken. This cannot prove conclusively or even semi-conclusively causality. Mendelian randomization and mediation and so forth are techniques that we are actually already using or planning to use, mostly in the analysis of the aging studies, where we have extensive genome-wide genetic variation information, and where basically we will use the genetic variation again as the instrumental variable in Mendelian randomization kind of studies to try to establish the direction of causality between several omics layers, and the endpoint, which is the clinical phenotype.
Okay. The next question comes from Larissa Constantino, who asks, what are the limitations of studying EL?
What are the limitations of studying EL? Well, one of the limitations is the one that I alluded to, right? Because when you're looking at extremely old individuals, it's hard to disentangle what is the signal that contributed to their longevity versus what is the signature that reflects their very old age, right? And so what is cause and consequence of their old age? So that's a big challenge. And so, yes, one of the things that we often try to do is to work with the offsprings of centenarian. So they're basically, you have offspring that are younger, they're often still in their 70s, but basically there you can compare, you can compare apples to apples, because you can compare offspring to control, which is basically age-matched individuals that do not come from families that have shown a predisposition to extreme longevity, exceptional longevity. So that I think is a big challenge. And of course, there are all the issues about limitation in sample collection and annotation, right? Basically, these extremely long-lived individuals may come from very different backgrounds, very different environments, and therefore being able to account for all the possible source covariates or all the possible hidden variables that might have contributed to their extreme old age is very difficult. But that's where actually approaches such as Mendelian randomization, mediation, and so forth can help actually point to what's really driving the phenotype.
Next question is from Gregory Dolinkowski, and he's asked, since a high-fat diet increases bile acids, does this mean that a high-fat diet leads to EL?
Well, that's what some people would say. I do not know. It's more complicated. Monounsaturated poly and monounsaturated fatty acids are important and have been shown to have a positive effect. But yeah, I do not know the answer to the question yet. Okay,
and given the time, this will be the final question, I think. Wait, so we have one more question. I'll try to get them both in here. This is from Zhendong Wang, this question, and he says he may have missed this on your last slide, but in your aging versus exceptional longevity study, have you investigated any correlation between the properties of plasma metabolites or any biomarkers which can be regulated by the gut microbiome of healthy aged people and long-lived people?
Okay, so this is a study that I'm not part of, but actually Paula and her team at Tufts, and also Albert Chai, who did the profiling, they are actually investigating the microbiome, but the results are not out yet, and so there are other studies, of course, not by us. They have investigated the microbiome and its association with age and extreme longevity, but the studies that are part of our team are still ongoing, and no results are available yet.
Okay, I guess that will, because of the time, we've reached the 1 o'clock. So given the time, that would have have to be the last question. I just want to thank you again for providing such a wonderful presentation today. Very interesting presentation. Thank you.
Thank you very much to everybody for listening. Apologies for any typos, quote unquote, and have a good day.